Welcome back to this next video, and this is the part 2 on the protein kinase A. In the last video, I've told you that kinases are enzymes which can add a phosphate group to their substrate protein, and we broadly divide these kinases into the dedicated protein kinases and the multifunctional protein kinases. Then I've told you that the specificity of a protein kinase that depends on the amino acid sequence that directly surround the residues to be phosphorylated, and this particular sequence was known as the consensus sequence. Then, to give you the importance of the protein kinase A, I've told you about some of the targets of the protein kinase A, and the protein kinase A can target different enzymes like the glycogen phosphorylase, which was very important in the fight or the flight response. Then I've told you about the protein kinase A targeting the different ion channels like the calcium ion channels in the cardiac muscles or the chloride ion channel in the small intestine. The protein kinase A can also activate the chromosomal protein like the H1 histone, which was very important in the stabilization of the nucleosomal structure and in the high order coiling of the chromatin fiber. Then I've told you that the protein kinase A can also go for the activation of different transcription factors like the Krebs, thereby stimulating the transcription of the responsive genes. Like one of the example was the tyrosine hydroxylase, which was important in the formation of a neurotransmitter, which was the dopamine. Then we started discussion on the fight or the flight response, and I've told you that during the fight or the flight response, the sympathetic nervous systems that signals the adrenal medulla of the adrenal glands, thereby releasing the epinephrine, which was responsible for the breakdown of the glycogen into the glucose and hence for the production of energy. Then we were talking about the pathway in detail, and the first component that we discussed, that was the epinephrine, which was the ligand in this particular pathway. And I've told you that epinephrine belongs to the catecholamine family, and it is made from the L-tyrosine through a series of enzymatic reaction. Then I've told you that if there is a ligand, there must be a receptor, and the receptor for the epinephrine was the epinephrine receptor, which is also known as the beta-2 adrenergic receptor, and which is also known as the G protein coupled receptor. And we discussed in detail about the structure of the receptor, like it have got the transmembrane domain, the extracellular loop, the intracellular loop, and then the carboxy terminal. And to this receptor, heterotrimeric G protein was attached. Then we were discussing about the attachment of the alpha subunits and the gamma subunit to the cell membrane, and I've told you that the phenomena that is responsible for this, that was the prenylation. And we discussed about the S-polymethylation as a subtype of the prenylation, in which we were adding the polymethyl group to the cysteine amino acid of the alpha subunit with the help of the enzyme polymethyl transferase. Now, in this particular video, I want to focus on another type of the prenylation, which is known as the N-myristoylation. Now, during the N-myristoylation, we are actually adding a myristoyl group to the nitrogen of the amino group of glycine amino acid. So, if this is a glycine amino acid, this one, if this is a glycine amino acid, and this is the amino group, what we will be doing is that we will be adding a myristoyl group to this particular nitrogen over here. Now, how this is achieved? Now, during this process, the enzyme N-myristoyl transferase, it utilizes the myristic acid, and it is going to attach this myristic acid, more specifically the myristoyl group from this myristic acid, to the to the nitrogen of the glycine amino acid. Now, one thing I want to clarify over here is that this N-myristoylation, it happens in the alpha subunits of a special family of the alpha subunit, which is known as the G-alpha-I/NOT family. So this is a special family of the alpha subunit. Now, in this particular family, if you look at the primary sequence of the alpha subunit, the first amino acid is glycine. Now, you may be thinking how the first amino acid can be a glycine. Usually, when the protein that is made, the first amino acid in all of the protein, that is methionine. But during the post-translational modifications, some of the amino acids, they are removed. And when you talk about the G-alpha-I/0 family members, in this particular family, during the post-translational modification, the first amino acid happens to be glycine. And if the first amino acid is glycine, you can actually attach these alpha subunits to the cell membrane utilizing the N-myristoylation phenomena. Now what you do is that you are using the myristic acid, and with the help of the myristoyl transferase, you are going for a condensation reaction in which you are removing this hydroxyl group over here. This is the structure of the myristic acid. You have got this CH2 repeated 12 times. When you have got these braces, and if there is a number outside these braces, that actually represent the number of times this particular structure is repeated. So if you look at this CH2 in the myristic acid, it is repeated 12 times. You have got a carbon over here with 3 hydrogen atoms, and then you have got this carboxylic group over here, this carbon with an oxygen over here, and with a hydroxyl group over here. So during the condensation reaction by the N-myristoyl transferase, this hydroxyl group is removed, and this hydrogen group from the amino group of the glycine, that is removed, and thereby generating a water molecule. Thereby you are going for a condensation reaction. Now what happens is, when you go for the condensation reaction, you will be attaching this carbon to this nitrogen over here, thereby making an amide link. So N-myristoylation is actually the addition of a myristoyl group to the amino group of glycine amino acid. As I've told you in the last video during the S-polymethylation, that we are not adding the whole of the palmitic acid, but we are rather adding a palmityl group to the cysteine amino acid. Same is the case over here. We are not adding the whole myristic acid to the amino group of the glycine. What we are adding is a myristoyl group. And what a myristoyl group is, the myristic acid without this hydroxyl group is known as the myristoyl group. And as you are adding this myristoyl group to the nitrogen over here, therefore this phenomena is known as the N-myristoylation. And this is you can say specifically happening in the alpha subunits of the G-alpha-I/0 family. Now another important prenylation phenomena that is happening in the gamma subunits of the heterotrimeric G protein is the addition of a geranyl group to the cysteine amino acids of the gamma subunit. If you remember in the last video, I've told you that the gamma and the beta subunits, they are attached to each other, thereby working as a single subunit. So if you are attaching this gamma subunit to the cell membrane, you are indirectly also attaching this beta subunit. So this gamma subunit is attached to the cell membrane with the help of this particular group, which is known as the geranylgeranyle group. Now this particular structure from here, this particular structure, that is actually your geranyl group, and what you are doing is that you are adding this geranyl group to the sulfur of the cysteine amino acid in the gamma subunit with the help of the enzyme which is known as the geranylgeranyle transferase. So we have discussed 3 subtypes of the prenylation. One is the S-polymethylation, the other one was the N-myristoylation, and now here the addition of a geranyl group to the cysteine the sulfur of the cysteine amino acids of the gamma subunit, and the addition of a geranyl group to the gamma subunit is giving hydrophobicity to the gamma subunit, and it can thereby attach to the cell membrane. Now, if you talk about this G protein, this G protein can actually exist in 2 forms. It can actually exist in the active form, and it can exist in the inactive form. Now, if a GTP molecule that is attached to the alpha subunit, it is going to turn it on. And if a GLP molecule that is attached to the alpha subunit, that is going to turn it off. So the activity of the alpha subunit of the G protein depends on the attachment of a GTP or a GDP molecule. So if you need the activity of the alpha subunit, you will be adding a GTP molecule to it, and if you do not need its activity, you are going to add a GDP molecule over here, and we will discuss that in detail how this convergence between the GTP and the GDP takes place in the next video. So if you like the video, do subscribe to my channel, press the bell icon and share it with your friends.